Today we have a lot to talk about. We're going to cover a little bit of morphology, ecology, some identification techniques, and we're going to go over some of the key mushroom groups that generally mirror the groups that Gary Linkoff established. Without further ado, let's get going. So I think Tom did a wonderful intro for me, so I don't need to say much more here other than I started Fungi Woman in 2018, and it's just sort of been growing ever since, and it has led me down the path that I am on right now as a, mushroom person, enthusiast, forager, and ended up becoming a book author, which kind of came as a surprise, but in my other life, I design software. So there's a little bit of overlap, but not that much. I certainly enjoy my mushroom career as well. Okay, so a little bit of baseline sort of terms, which I am sure everyone is familiar with. So what are fungi? They are eukaryotic organisms that are heterotrophs, which means that they cannot make their own food, which is what differentiates them from plants. They also have chitin in their walls, which makes them a lot more related to insects than plants, and they reproduce through spores. So those are kind of the biggest differences for the kingdom fungi. And they are known for playing an important role in nutrient recycling, as you all know. But what are mushrooms? So mushrooms are the fruiting bodies of certain fungi that typically appear above ground, and they contain the reproductive units. So a popular metaphor really is a mushroom is kind of like an apple. So common misconception often is that if a mushroom is picked, it's destroying the organism. It's not really true. The organism is actually the mycelium. It's not just that showy kind of fruiting body that's above the ground. And not all fungi produce mushrooms. I think the current estimates are about 12,000 species in North America, and Pennsylvania or Ohio is home to about 3,000 different kinds of mushrooms that we know of based on sort of tracking. Mycelium. Mycelium is really where the magic is. It's the underground vegetative part of the fungus that extracts the nutrients. And it is sort of the root-like structure that consists of the mass of branching thread-like hyphae. And this hyphae forms this massive underground network that connects with the roots of trees and plants and has been sometimes or recently referred to as the wood wide web. And it's been posited that not only does it allow for the exchange of nutrients and water, but also that chemical signals, which can somewhat be translated as information, can be passed along this network. Now, since this concept has come about, there have been some I think pushback on that. So it is still an area of research. You might have seen articles that say like fungi and trees can talk to each other, and it's just not really quite true. Because also, as like Merlin Sheldrake says, using sort of human concepts to describe what fungi do is kind of inaccurate. So we're still researching this, but if you hear this term, it's that's sort of what it refers to, and it has been sort of explained slash proven that there is some communication happening by Dr. Suzanne Simard, who authored the book. I believe it's called the Mother Tree, and it's a great book, so I highly recommend that. Okay. So very briefly, life cycle starts with a mature mushroom that drops spores that sort of land in places. And then when the conditions are right, they sort of sprout into a singular hypha, which then fuse and then form a mycelium. And then again, when the time is right, a dense knot of hyphae called a primordium forms, and that expands and creates a small mushroom that's often called a button, and then sort of the cycle kind of happens again. So that's generally the life cycle of a mushroom. So how many species are there? I have heard some varying estimates. I think the more conservative ones are 2.2 to 3.8. Some of them go all the way up to 6.2. I think I've also heard like 9 million thrown around, but we don't really know because a lot of them are undescribed, and we currently have about 155 of them described. And apparently, we describe a lot of new species every year, thanks to also now DNA sequencing. But unfortunately, we're not going to be able to describe them all because of the loss of habitat and all the host loss that is resulting from sort of polluted environment. A lot of those species will likely become extinct before we even know that they were there. So that's why it's so important to record our observations, to document the microdiversity around us. And I know that your club is doing that. And I love the segment on like the INAT, spotlighting the INAT finds. That's really great. So how can we classify mushrooms or fungi? There's four ways that I usually talk about. One is the taxonomy, which is sort of the scientific way based on genomics and phylogenomics. We can also look at fungi in terms of ecology and how they interact with their environment. Lots of people love to categorize fungi based on whether you can eat them or not. It's kind of one of the more useful ways of talking about mushrooms. And then finally, morphology, which is where we're going to spend a lot of time today. So, based on taxonomy, all of this is sort of based on the sexual reproductive structures. And there are some major divisions. Like mycologists, scientists, academics still can't really decide on how many of these we have. Some have 4, some have 5, some have 9. I think this is the one that's like most commonly accepted. And usually the ones that, mushroom clubs and sort of amateur mycologists kind of focus on are these two, which are often called the higher fungi, which are the Basidiomycota and the Ascomycota. And the biggest differences are in sort of their reproductive structures. So the Ascomycetes, or the ascos, as some people like to say, usually have 8 ascospores that are sort of in these sacs, whereas Basidiomycetes have basidia, which are, again, microscopic reproductive structures that usually sort of the spores hang off of these clubs, which is also called club fungi. And there's usually 4 of those tiny little projections. And they have also different like mechanisms for the dispersal of their spores. So that's why they are sort of separate in these two groups. Okay, so ascos is the largest division of fungi. The recent estimates are about there's 93,000 species that have been described. There's familiar examples, I'm sure, to all of you. They include the delicious morels, the false morels, also the dead man's fingers, like the Zillaria genus. It also includes the cup fungi that we find usually in the spring. So many of these are edible. They are, you can forage for them. Many are beneficial to humans as sources of sort of medicinally important compounds, including antibiotics. It's also thanks to the ascos that we have bread and cheese and beer. So lots of awesome organisms in this group, but a lot of them are also pathogens of plants, animals, and including ourselves. The Basidiomycota is also a large and diverse division. It includes sort of what we think of the typical mushrooms. And some people, I recently learned, only reserve the term mushroom to basidiocarps, which is why we sort of call basidiocarps versus ascocarps for these specific divisions in the fruiting bodies that are produced by fungi in these divisions. There's varying opinions on whether the term mushroom is reserved just for this division or not. Um, so what's in this division? It's all the delicious things like chanterelles, boletes, the polypores, puffballs that you talked a lot about today. And some of these mushrooms may have medicinal properties. It's currently under research. And many of them are used for other uses that are currently sort of being researched and trying to be figured out, such as restoring the environment through mycoremediation or mycofiltration, or using their mycelium for like mycotextiles or producing furniture and stuff like that. So lots of potential in this from this division. Okay. In terms of ecology, there are 6 ways we can sort of group fungi. So the first is mycorrhizal, which is the mutually beneficial relationship with plant roots. So those mushrooms, those fungi are usually terrestrial, and they have to make those connections with the tree roots. Otherwise, they cannot exist, and actually vice versa. Parasitic fungi are the ones that attack living trees, and they cause disease and eventually death. And the next group is the saprobes or saprophytes, which grow on the dead organic matter. So those are the nutrient recyclers, and they decompose the cellulose and lignin. And if we didn't have them, we would literally be buried in organic waste. And fungi are super cool because some species actually leverage all 3 of these ways of living and obtaining nutrients. For example, the popular hen of the woods sort of starts as a weak parasite, but then if the tree dies, it continues as a saprobe. So I think morels are also, sometimes you'll see descriptions where, possibly mycorrhizal. So these fungi like grow on wood and they decompose the wood, but there also has been observed some relationship to specific trees or plants as well. So lots of cool ways to live for fungi. And the 3 other groups are the endophytic fungi, which live inside plant tissues without causing harm to the host. They're actually very beneficial, and plants, turns out, cannot live without these endophytic fungi. But they are microscopic, so we don't really see them. We also have lichens, which are the symbioses between algae or cyanobacteria and a fungus. So those are visible. They grow on rocks, also on the ground, on wood. And then finally, we have aquatic fungi that live in aquatic habitats, and they're kind of mostly microscopic. Now to the fun stuff. So I like to think of edibility as a spectrum. So on the one end, you have the deadly ones, which can kill you, which there's actually not that many of them, as many people find surprising because, I mean, here we love fungi, we love mushrooms, so we know, but to the general public, whatever I say, like, there's actually not that many of them. They're very surprised. And my advice always is, to know the deadly ones in your area first before you start foraging for any other ones. Then we have poisonous mushrooms that will make you sick, but likely won't kill you. They will just cause some headache, gastrointestinal upset, vomiting, diarrhea, nothing you'd want to experience. Then the next group is inedible mushrooms. So mushrooms are considered inedible for like multitude of reasons. Either they don't taste good, or their texture is too kind of tough to eat, or they are too small and flimsy to actually like bother with. But if you were to eat them, like nothing would likely happen. But they're just usually not considered edible. The next group is what I call conditionally edible mushrooms, for which you have to meet certain conditions to be able to eat them. So those mushrooms are morels or chicken of the woods because they contain sort of mild toxins when they're raw or undercooked. However, these toxins are not thermostable, which means that they are destroyed by cooking. And when you cook them long enough, these mushrooms, they are safe to eat. Now, that is not true for this group over here on the left. It is not true that if you cook a mushroom, you can safely eat it. No, not true at all. And then finally, you have the delicious, delicious ones, which are like the chanterelles, the king boletes, that are edibles, or sometimes they're called choice edibles for like this especially delicious ones. Okay, before we actually get to morphology, I want to pause if there's any questions or comments that anybody wants to discuss.
Yeah, nothing's come up in the chat yet.
Okay, great. Okay, so quick overview. I'm sure I'm not telling you anything new. Often when we think of mushrooms, we sort of think of the stipe, we think of the cap, we think of the gills or the pores underneath. And, maybe there's something on top of the cap, like warts or patches and things like that. But you all know that mushrooms do what they want, and they have evolved into a multitude of shapes and structures. So here's just a few of them. And we're sort of going to go one by one and talk a little bit about each group. So I'm not going to get into it too much here. But when we're talking about morphology, you sort of have to train your eye to be able to kind of distinguish some of these characteristics and things that we want to talk about when we talk about mushrooms. So there's different ways we can talk about the cap shape. There's different terms for that, and they each mean something very specific. And the same thing with cap margins, like they can be scalloped, or you can have these striations, these tiny sort of leftover edges or indents from the gills. You can have a cracked cap margin. There's different types of cap textures that you could encounter when looking at a mushroom. We also oftentimes talk about cap margin cross section, whether it's incurved or enrolled or plane, all these fancy terms. And when we talk about the underside or the hemenophore, there's different structures underneath. So the gills and the pores are probably the most well-known. But, for example, chanterelles, those have ridges. And the hedgehogs have teeth, sort of these like tiny little icicles kind of hanging off of that cap underneath. Or crust fungi have a smooth underside completely. We also notice gill spacing all the way from sort of very widely spaced, which we call distant, to like crowded, where the gills are super, super close to each other. And then a lot of times you will also hear the gill attachment, which I personally find the most challenging to be able to determine, unless it's between like free and attached and maybe decurrent. But sinuate, I don't think that I honestly, I don't know that I would be able to tell. But those are all the terms that are out there that are good to start noticing and start being able to differentiate as you're sort of out there in the field. We have more things. We also notice pore shape. Um, so angular, like the hexagonal pored polypore has very distinct pore shape. When we talk about boletes or amanitas or any other mushrooms, the stipe shape is super important as well. Like, does it have a bulbous stipe, or is it just sort of swollen, kind of like that, or does it have a rooting one? And then finally, we also look at stipe texture, which there's different terms. I think one of the biggest ones that we talk about often is reticulation, which is sort of this network on a lot of boletes, or perhaps the scabers that the scaber stalks have, or, amanitas have rings, or there's also a ring zone that you can notice if once the ring is gone. So lots of tiny little things to notice about one particular mushroom when you're sort of looking at it. Okay, any questions about those or comments?
Nothing yet, Barbara.
Say that again, Tom?
I said nothing yet.
Nothing yet. All right. So we'll keep going. So Agarics is sort of the first big group, which is a large group because it includes all the gilled mushrooms. So if it has a gilled underside, we can call it an agaric. And that's it. It's just the single morphological group based on that one singular characteristic. Oftentimes, identification in this group can be difficult because it is such a diverse group. And many of those can be frustratingly similar. The species in this group generally range from delicious to deadly poisonous, and they can also be psychoactive. So the agarics are usually not the ones to start foraging with. They are sort of a more of an advanced mushroom to be able to safely eat because, as Cindy mentioned, you better make sure that you 100% know your friend before bringing them to your table. So those are not beginner mushrooms, but there are some groups that are easier than others, like milk caps, for example, that will sort of bleed latex. That's a pretty clear characteristic. But yeah, there's a lot of them, and some of them, especially if they're brown, can be very difficult to identify. Amanitas are also agarics, but they're also amanitas. It is a large genus that contains approximately 600 species that's currently divided into 7 specific sections. So that's just one way of kind of grouping related species together. And they include some of the most toxic known mushrooms found worldwide, including the biggest culprit of the group, which is Amanita phalloides. And that is currently invading and overtaking California in massive, massive numbers. I believe Dr. Pringle is researching that quite well. One of the common mushrooms that is on the East Coast here is the Amanita flavoconia, which is a beautiful Amanita that has sort of yellow coloring, and it appears sort of early to midsummer. And of course, it includes like the mushroom, the Amanita muscaria, which is the well-known sort of red cap with white warts and sort of cog-like sort of rings at the bottom above the base there. Uh, as a genus, they are responsible for approximately 95% of the fatalities, according to the MAW toxicology report. And like I said, the biggest culprit of them all is the death cap, which is responsible for about 50% of those poisonings. The most potent toxin present is the alpha aminidin. It's not actually the muscarin, which is what the muscaria is named after. Um, yeah, so those are amanitas. Again, they do contain some edible mushrooms as well, which is the blusher, Amanita rubescens, which is one of my favorites to eat. But it is certainly one that you have to know very well to be able to eat that safely.
The
Bullets, yeah,
we did get a question.
okay.
Alan asked if that 95%, is that fatalities in the US or globally?
I believe because it is a NAMA, I believe that is just North America because that's the source. It's the NAMA toxicology report. But I am like 99% sure that that is true. Um, oh, the woman in Australia fed her guests. You know, I've heard of that, but I actually don't know if she fed him the Amanita phalloides, but probably. Because that, I mean, that would be the best choice, I would think, if that's what you're trying to do. Um, yeah. All right. So we'll move on to the boletes. So boletes are the easy ones if you're starting to forage, because on the East Coast, we don't have a lot of the Rubroboletus genus, the ones that are very poisonous. They appear sort of in the Rockies and out west. But, you know, that doesn't mean that they are not here. Like just because we haven't found them yet doesn't mean they're not here. So I wouldn't necessarily say flat out that we don't have any poisonous boletes in Pennsylvania, although our bolete specialist Scott does say that. But he also says, as I'm repeating him here, that just because we haven't found it yet doesn't mean that it's not here. So caution is always advised, but boletes are easy to identify. They sort of have this like spongy surface with the pores, and which makes them sort of easily identifiable. And they're usually very meaty mushrooms, which makes them um, very appealing to put in your basket. Many of them are the delicious edible species that you're looking for, but there are some, like I said, that are potentially poisonous. But all of the ones that you see here are edible, and all of them do grow here in the Northeast. With very few exceptions, boletes are largely mycorrhizal, just like amanitas, and they have to make those partnership with the tree roots. And they do grow both in the forest as well as in parks. I regularly find boletes on my way to work in late July. Um, and so yeah, they grow both in sort of forest and urban environments. This is the scaber stock. There's sort of different groups within boletes that we kind of talk about. One of them is the scaber stock, which sort of has these like dark, kind of like scabers. It's kind of like almost like if you were to take a paintbrush and kind of painted the stalk a little bit with it. And they're usually brightly colored. They either have like brown or red or orange caps. In the Czech Republic, they're actually a more exciting find than the king bolete, which is very common in the Czech Republic. We have a lot of spruce and oak there. But these Leucinums, those are the ones you want to find because they're just so pretty. Chanterelles is another popular group of edible mushrooms. It sort of includes like chanterelles and trumpets. So a trumpet is the Cratylus phalanx here, up here, and then the here, Cratylus tubiformis, which grows here as well, specifically in Cook Forest is where I found them. And the difference between trumpets and chanterelles is that when you kind of slice them in half or tear them in half, the trumpets will be hollow, whereas the chanterelles will be solid and kind of white and stringy on the inside, especially the golden chanterelles. So, and they are one of the easiest ones to forage for because they don't have gills or pores. They have these wrinkles, which can sort of range from very pronounced to just kind of barely visible there. I'm sure you have probably found some smooth chanterelles out there in your woods, and sometimes those are completely smooth on the underside. They're also morphologically very variable, but at the same time, they are quite distinct that they are a great beginner mushroom as well. In Pennsylvania, we usually get them at the end of July. That's kind of when we kind of get like that big flush for the golden chanterelles, and then the Cratylus tubiformis is usually a more of a colder season, like more towards the end of the fall. We do find them during the Garilinka end of September, if the weather is good, if there's rain. And then the black trumpets are quite possibly some of the most delicious mushrooms you can eat. Actually, when I first found them in the Czech Republic and I cooked them, I overcooked them and they tasted like cardboard. And I was like, I don't know what everybody is like talking about. Like these mushrooms are disgusting, I don't know what's up with this. And then I actually read up on it and I just totally destroyed them. So the next time I cook them, I cook them right and delicious, delicious, delicious. So if you have these spots, guard them with your life and keep returning back because those are some great mushrooms there. Okay. Polypores. Polypores, I'm sure you have seen around. They are quite common. They can form these large fruiting bodies. They can also be a bit more tiny, like the turkey tail, for example. Or you could get sort of massive brackets like the hemlock reishi, the Ganoderma tsuge, which grows with hemlock. And the chicken of the woods can definitely make sort of large brackets as well. So they range in size, but they are all saprobes or parasites. Those are not mycorrhizal mushrooms. They grow on wood and they are the nutrient recyclers, or parasites. They attack sort of still a living tree. They're also called like bracket fungi or shelf fungi. And the ones that are sort of these woody kind of, they grow year after year, are sometimes called conchs as well. One of the more common ones is the Fomitopsis species, which we have, it's quite common in Pennsylvania. It's the red-banded conch, or red-banded belt conch, sometimes what am I looking for? I think it's called the red-banded conch is the common name as well. But it'll have this red band on the outside, which is kind of the freshest part, the most recent part, because these are perennial conchs. They will sort of add another layer every year. Crusts can sometimes be similar to polypores. So for example, the Sterium species is a very common crust that's common here. And it can sometimes resemble the turkey tail. But the biggest difference is that crusts are generally smooth on the underside if they do produce sort of these bodies as well. They can also be completely pressed to the substrate, like the giraffe spots or the crowded parchment, which just sort of hugs that branch or that log, and then when it can, it'll actually create those tiny, tiny little shelves to kind of help with spore dispersal. Other crusts can have wrinkles as well, but generally they're kind of these decomposers that spread along these woody surfaces. The fancy word for it is resupinate. All right, corals and clubs is another big sort of morphological group. It is a catch-all category for anything that looks like a coral or a club. So these fungi are not related whatsoever. It is just based on that their shape. So they can range from sort of these single clubs to these spindle-shaped sort of clustered structures, or they could be highly ornate, like the crown-tipped coral that is usually grows in early summer. And one of my favorite things about these fungi is that they can be so colorful. So colorful, and they're so pretty. It literally looks like a coral from under the sea. And one of the most recent developments, thanks to DNA testing, I don't know if you've heard, is it turns out that the purple coral right here used to be called Clavaria zollingeri. And it turns out that, and it has been assigned a vulnerable classification from the IUCN Red List worldwide. But turns out that we do not have Clavaria zollingeri here in North America. Clavaria zollingeri is a European species that has the vulnerable classification. And here in North America, we have two different species, Clavaria amethystina, which seems to be quite common actually in places in some years. And the other one is Clavaria, oof, there's another one. Viola, oh, something. Give me a second. I want to tell you that exact name. Is Violacea pulchra. And they are morphologically quite indistinguishable, apparently, except the other one, not the amethystina, uh, supposedly fades to pink a little bit. That's like the only way to tell them apart. But pretty interesting research because that helps us update our conservation rankings and understand a little bit better what these fungi how these fungi live and how to best protect them. Okay. So another group is jelly fungi, which are definitely a lot of fun to touch. As you can see, this one here is a jelly fungus, but it also looks like a coral. So really it could be present in both groups because it is still a jelly fungus. So this is the Calycera viscosa, which grows on decomposes conifer wood spruces, most common. So A lot of jellies have this foliose, irregularly branched, gelatinous fruiting bodies. Um, sorry, Zoom is always in the way. Um, and they're very rubbery and sometimes kind of sticky to touch. They often grow on in colder months, so you will start to see them. You have already observed the Exidia crenata. And Tremella mesenterica, the witch's butter, is also very common. And of course, the sought after wood ears, which is Auricularia species. This one is the European one, Auricularia judae. Here in the US, we have two. We have the Americanum, which grows on conifer, and then we have the Angiospermarum, which is on hardwood. Many of these are edible. They usually don't taste like much, but they do have a good texture. And a lot of them do really well in Asian dishes like hot and sour soup or stir fries or sort of Asian salads. And this one, the Exidia crenata, I like to call woodland raisins. And they like to jump out of your pan if you try to cook them. It's actually quite fun trying to like keep all of them in the pan. Um, so, and this one is just, I mean, you can see it everywhere. It is very common. Tooth fungi are super photogenic as well. They're also called hydnoid fungi. It is another group that is just based on the reproductive structure, which is the pendant tooth-like or spine-like projections where the spores are. And cool thing is that the teeth are always oriented so that they will be exactly perpendicular to Earth. So usually the teeth are actually kind of like this. They're not like a cylindrical thing. It is a tooth that's kind of like that. And so then the spores kind of drop easily from that. It's not, again, not a natural taxonomy group. It turns out that many different fungi have evolved this particular reproductive structure. So it's kind of the same thing. They took different trains, but arrived at the same destination. Some of these are edible. Hedgehogs are related to chanterelles. They're very delicious. Some of them are invasive species, like the Rhodolomyces coplendii, and they're not edible. Some of them are just super cool to photograph, like the toothpick fungus. Some of them are also jelly fungi and tooth fungi at the same time, like the cat's tongue in the top left here. And of course, Hericium, whether it's Coralloides or Erinaceous, delicious, delicious mushroom that is sort of reminiscent of seafood, both in taste and texture. So,
Question for you, Barbara. On the
yeah.
last slide, Scott asked, are there any inedible or toxic jellies?
Yeah, so not that I know of. And when I was doing my research for the book, I did not find any poisonous jelly fungi. Now, that doesn't mean that, they're not out there. Um, I'm not aware of any. I'm not aware of any research that identifies any.
And of course, there's edible, and then there's edible and enjoyable.
Yeah, exactly. Like, so the wood ear, that's an edible, that's a commonly edible one. People actually forage that and eat that, and that's a common one to forage for. The witch's butter, this faux tremella, and the Exidia crenata, like the amber jelly roll, those I think I would call like you kind of have to be into mushrooms to like forage for these. Um, the Tremella fusiformis is a common edible and medicinal mushroom. So that is definitely one that is well known. You can also buy this dried in a lot of Asian stores. And you will get so much of it. Like it just poofs up like insanely. And this one, the Calicera viscosa, like while it's not, it's not poisonous or anything like that, like it is a common one in the Czech Republic. I will sometimes see like weird reports of people collecting it and kind of putting it in with their like pickled other mushrooms because it kind of looks cool. So it can be used as like garnish, but it's not really, commonly foraged one. So good question. Okay. Cup fungi, super cool too. Lots of colorful members here in this group too. Lots of tiny ones as well. Of course, the elf cups, the turquoise or green elf cups are super tiny, like sometimes just barely measuring 3 millimeters across. It is another sort of large group of fungi that is spread across multiple families and genera in the Ascomycota division. So, and again, this group is solely based on the shape of that fruit body. So a lot of them will be brown, but like I said, lots of them are very colorful. You have the scarlet elf cups, the Sarcosypha species, which are popular spring fungi. This one is again like this in particular has been popularized in social media in recent years. It doesn't really taste like much. I've eaten it twice, I think, in my life, because I too fell. I just prey to the social media train, and I was like, I have to make my own Scarlet Elf Cup pizza. And it is really cool. They're actually quite good, and they are very effective on that pizza um, instead of pepperoni. But the one thing to remember here is that these are some of the first fungi, fresh fungi, to appear after winter, and they do support a lot of animal communities. After the long winter. So it's important to share this one and not forage all of it when you kind of see it out there. None of the other ones are traditionally edible because they're just super tiny. But I also I'm not aware of like the brown cup, for example, to be poisonous, but it's also not one that like people forage for. They do have an awesome spore dispersal mechanism. If you blow on them, um, you will disturb them and then they will sort of put this spore cloud out. There's a lot of videos out there that kind of capture that, and you can actually observe that in the wild. It does take about maybe like sometimes like long time, like 5 seconds, like you'll blow on them and then nothing will happen. And then suddenly there will be this like poof of spores in front of you. So highly encourage you to try that out. So yeah. Um,
Yeah, we had another question
yeah.
because we have such a color palette on this page.
Yeah.
Why did some fungi evolve color? Now,
Oof.
I've heard a couple theories on this, like brighter colors to reflect more solar radiation, darker colors to absorb more solar radiation, but I haven't heard any
Mm.
definitive theories. I don't know. Do you have anything more?
No, I haven't. The only thing I sort of researched that in relation to was the turkey tails, which is partially based on genetics and partially based on the substrate that they are decomposing slash growing on. So that can affect their color a little bit. But that's all I've heard. So yeah, it's sort of the same question as like, why are some fungi bioluminescent? I don't think we have the answer for that either. Um, Not a definitive one anyway. So yeah, it would be interesting to know. All right. Morels and allies. So all the false morels and all the thimble caps as well. So the true morels. So that's the Morchella americana, the diminutiva, the punctipes, which is the half-free morel. So all of those will be hollow when you sort of slice them open, and they have this honeycomb sort of appearance to them. The false morels, as sort of a giant group, is the top one, the Neogyrometra caroliniana. And actually, I think this is, I can't remember. They just reclassified them. I don't think this is Gyromitra corfii anymore. I think this is something else. It's like a really weird genus. Um, Hold on, I'm gonna look that up because I do want to tell you. Um, Does anyone know? Oh yeah, here it is. Maublancomyces. That's a mouthful. So that's another sort of recent development in this sort of group. They split Geromitra into multiple genera. So that is that. Okay, let me get this back. Okay, you seeing this? Okay, and the question is also always about, can I eat false morels? And the answer is it depends, because it very much depends on that specific species. So again, it just sort of goes back to needing to know the exact species that you're bringing home, because these sort of generalized answers are not possible. So for example, the Neogyrometra caroliniana is edible if you cook it well. Same thing with the Maublancomyces corfii. Those have been consumed by people, but again, they are conditionally edible. You have to meet certain conditions to be able to eat them safely, just like morels. And the verpas, the Verpa conica, those are also edible. They're not as common as they are here, compared to like the Czech Republic, for example. But they are edible as well, conditionally edible. And this is one of the ones that is possibly saprobic and mycorrhizal at sort of different points in their life. So kind of cool there.
If it's okay, I'd like to add just one
Yeah,
more point about the
yeah,
false
absolutely.
morels, the
Yeah,
Gerometras. So, just for informational purposes,
yeah,
the Gerometras contain a toxic substance that's
yep,
called gerometrin,
yep,
and your body metabolizes that to a substance that's called monomethylhydrazine, which
yep,
is a component in rocket fuel and highly toxic. It's
yeah,
volatile, so it can be cooked off, but
yeah,
yeah, so it's
yeah.
it's
Yeah, yep, 100%. Although that's I believe true for the Gyromitra esculenta. The Neogeromitra does not contain sort of these like massive levels of the gyromitrin. So when you're cooking the Gyromitra esculenta, which is highly, has been consumed for centuries in like Finland and sort of these like Nordic countries. Um, it's recommended when you actually boil this that it's like a 3 times boil. But if you're inside, you need to wear like a KN95 so you don't breathe in the fumes, and it's just kind of like a lot. But actually, because that toxin is not thermostable, it'll get either cooked off, or actually the way they consume them in these Nordic countries is they dry them first, and that sort of degrades the toxin as well. and then they can be sort of safely consumed. But you know, these things can accumulate in your body as well. Different people kind of metabolize the different things in the different toxins as well, which is why there were these like surprise poisonings. So this group is problematic to say the least. So do your research before you consume them. But there is a really good paper that is new that summarizes all of this, and the paper that split this genus actually ties it back to the toxicity. So it's really good. So I recommend looking that one up. Okay. So the next one is, I think this is our second to last, puffballs and allies. So again, those are sort of these ball-shaped mushrooms that are unique because they produce spores inside their fruit bodies. Generally, we group this or subdivide this group into 5 major groupings, which are the common puffballs. We have the giant puffballs. We have the earth balls, which are the sclerodermas. There's also stalked puffballs, Tulostoma. I have yet to find one of these. And then we have the awesome earth stars, which kind of split and produce these very exciting shapes on the ground. Most of them are terrestrial, but they are actually decomposers. And the common puffballs, the Lycopterodon species, or you know, the Epioperodon, the pear-shaped puffballs, are awesome beginner mushrooms because they are easy to identify. And compared to the earth balls, the Scleroderma species, the puffballs will also have more of like a marshmallowy texture, as opposed to the earth balls are a lot sort of harder. And that's your additional kind of characteristics besides just slicing it in half, and the puffball will be white and the earth ball will sort of be purple or black or anything like that inside. Yeah. And finally, we have the stinkhorns, which are species that are primarily found in the tropics. However, we do have some. We have sort of the lower 3 here. The Phallus imputicus, the Pseudocolus fusiformis, and the Phallus ravenellii. Those are the common species that you can encounter in the Northeast. They are generally very unique looking, and they have these very unique spore dispersal mechanism, which relies on odor, such as rotting flesh, and that attracts the insects, who then land on those mushrooms, like the Clothris archeri. And then those flies, those insects get that black goo on them, which is the spore mass. And then as they kind of fly and move around, they sort of help disperse the spores. These mushrooms begin their development as these oval structures like eggs. And a lot of those eggs are considered a delicacy when they are in that stage. I have not had the pleasure to eat those. Or maybe I should say I am not brave enough to eat those. But yeah, apparently very delicious mushrooms at that stage. And of course, they're so fun to photograph. A funny story about the Clothris archeri here, the octopus stinkhorn, which does not occur here in North America, but is an invasive species in Europe. My mom one day, like, sends me this text and she's like, oh, there's these like really weird mushrooms growing like behind the summer house. And, you know, they're like really cool. They're like these red and it kind of looks like an octopus. And I'm like, oh my God, like no way. And then she sent me a picture and I was like, oh my gosh, this is only on like my top 10 list of mushrooms to ever photograph. And they just so happened to be behind our summer house. But this was in early July. And actually, They did continue to fruit through that whole summer. So when I arrived in August, I was actually able to take photographs of them. And they haven't fruited there since. And our theory is that we bought some wood that we were chopping up and kind of using circular saw to like prepare for winter. And there was a lot of sawdust that remained on that meadow. So we think that's what happened. But they haven't occurred there since. So that was that. Okay, so off to our next and last section. So how do you actually identify all these mushrooms and what do you all need to know? And what is generally the order that we employ when we sort of begin mushroom or fungi identification? So number one is the habitat, right? Like where is it growing? It's also the picture you kind of want to capture for your INAT observation. So is it growing in the woods? Is it growing on your lawn? Is it growing in your flower bed? Is it growing in the desert? All sorts of things like that. So that's number one. Number two is the ecology. So how does it grow? Is it growing on wood? Is it growing terrestrially and therefore it's likely mycorrhizal, or is there like wood hiding underneath, buried, stuff like that. So you want to determine that. Your next one is the morphology. So it's all those like fancy term, the mycologies that you want to notice in terms of the stipe, shape, texture, gills, gill spacing, gill attachment, cap texture, cap shape, cap margin. All of that you kind of want to notice and consider when you're trying to identify a mushroom. The fourth technique is your other senses, smell and taste. Some mushrooms are like the anise, the Clitosiba odora, which smells of anise, which is really cool. There's mushrooms that smell like beef bouillon. There's mushrooms like the stinkhorn that smell like rotting flesh. And sometimes also being able to taste a little bit of the mushroom at the tip of your tongue and then spitting it out can tell you something. This is specifically important, for example, for russulas, brittle which if they are peppery, generally they're considered not edible. And if they are mild tasting, they are edible. So that's, for example, one group where taste can become handy. Now, you can actually safely taste any mushroom, even the deadly ones, in theory, because mushroom poisoning happens through digestion. But you should not do that. You should absolutely not try to taste Amanita bisporidea or the Eastern Destroying Angels by any means. So before you kind of employ this technique, it's good to know which ones are potentially poisonous and not taste those. Okay, last one that is kind of generally accessible is the spore print. So if you sort of take your mushroom home and you put it on a piece of paper or aluminum foil or glass, and you let the spores drop, it's usually good to actually cover it with another bowl or something to prevent airflow. Then you can observe the color of the spores. This, for example, is particularly useful trying to differentiate a bluet from a web cap. So a bluet, the edible one, will have a white spore print, whereas the Cortinarias, the web caps, will always have rusty spore prints. And you certainly don't want to eat any web caps. So that is a useful technique for that. Now, these other ones are a bit more advanced. You can also use chemical reactions to kind of help you with some IDing characteristics. There are chemical kits out there that some mushroom clubs sort of give out or you can get. But they're kind of mostly kind of fun to do. And then lastly, you can of course leverage your microscope if you have one to observe some of the structures under the microscope. And finally, DNA analysis is the last sure way to identify mushrooms, which has recently become a lot more accessible to mushroom clubs who sort of partner with some of the national labs like Stephen Russell's Mycota Lab to kind of help figure out new species, which is kind of exciting. Okay, and I think that's it. So yeah, if you're interested in my book, it is available on Amazon. There's a whole bunch of recipes and sort of in-depth profiles on mushrooms and also sort of these chapters that are organized based on kind of the groups that we talked about. So that's it. Thank you.
Thank you, Fungi Woman.
Yeah, thanks. That was a lot. I feel like we just kind of zip through that, and this is of course just like barely scratching the surface for these groups. So
I think we handled most of the questions as we were going through it. The only one that I thought we'd go back on was somebody was questioning the Amanita. You mentioned that Amanita rubescens was
yes,
Amara rubescens is potentially edible, and I kind of gave a answer about that. That I don't
yeah.
really, I
So
don't really think people should be eating it, but
no.
of course opinions vary.
Yeah, opinions vary. So there's different, this is a European species. So this is the Amanita rubescens. The North American species actually is a little bit more brown, but the flesh is still turns pink and sort of does blush. Um, the Western species is Amanita novinupta. And here, that's the one that has been described, but currently, I believe, under the complex of Amanita amerirubescens, there's 9, I think, cryptic species in there. They're kind of waiting to be described and all that. So, yeah, I have, yeah, I mean, I eat these in the Czech Republic regularly, but that's because my grandma foraged those, my mom foraged those, and I foraged those. And yeah, you kind of develop an eye for them, but it's certainly not a common edible. I think Alan Burgo, the forager chef, also eats them. Yeah.
Yeah, and I've heard on the other side of the spectrum, I think it's David Aurora will
Yeah.
cook amanitas for groups of
Oh,
like
yeah.
100
Like the
people.
muscaria.
Yeah.
Yeah.
Mm.
Yeah. I mean, I don't think that's needed, but
Mhm.
apparently they're delicious. And, again, it just kind of, I think, depends on your risk level and, yeah, and how confident you are in your ID.
Well, I think we have exhausted the questions. I would like to thank you again. This was
Yeah, thank you
fantastic. We're
for
getting all kinds of platitudes in the chat
having
box. I'm
me.
sure you're seeing.
Thanks. Yeah.
Yeah. All right. And look at the time. We're going to finish 5 minutes early. How
Yeah.
rare for us. Okay. All right. Well, thanks again, Barbara. Thanks
Yeah.
again,
Thank
everybody.
you.
Thanks
Bye.
for coming out.
Yeah.
And we'll be seeing everybody on the Mushroom Trail again soon.
Yeah.
Good
Bye.
night,
See
everybody.
y'all.
Bye.